On this boat, we want to sample both the fish community and the water that the fish are living in. So to do that, we're catching the fish with a large trawl net that's about 40 meters wide by 15 meters deep, and we fish it at different levels so we can see who lives where in the water column. This is a collaborative program between DFO, the Pacific Salmon Foundation, and the Pacific Salmon Commission. So we're doing a number of different studies while we're on the vessel. The work on juvenile Chinook salmon is a collaborative project with the Pacific Salmon Foundation. We've been conducting this survey for about 15 years now, since 1998. And from this survey, we can actually estimate the abundance of the juvenile salmon, and we can take a look at their size and their distribution. We want to know how big they are. We're measuring length and weight.
1, 2, 3, 2, 0.
So behind me, Carol is looking at the diets of these juvenile sockeye salmon, and she's identifying the volume that's in their stomach and the digestion rate and the major prey groups that are in there and the proportion of each prey group that's in the gut contents.
I see euphosates in there, and I see hyperids in there.
And that's being recorded by Lenora off to my left. Mary is taking some of those gut contents and freezing them in bags for an RNA-DNA ratio analysis that will be done later on in the laboratory. We're taking muscle tissue to take a look at fatty acid composition. We have somebody on board who's taking blood samples, and that's to sample for IGF. We're removing the otoliths from the fish. One of the structures we remove from the salmon is their otoliths, or their ear bones. And they're just a small... A number of the fish are being sampled for Christy Miller's lab at the biological station. And what the girls are doing in that lab is they're removing small parts of the tissue, so the brain, the head kidney, a piece of the liver, some of the gut tissue, some muscle tissue, to do some molecular work back at the biological station. When the catch comes on board, sometimes we'll catch other species like dogfish, pollock, lamprey, anchovies. There could be a number of different fish. If the fish are alive and they come on board, we measure them and release them immediately back into the ocean. To sample the actual water, we use two different pieces of equipment. The first is called a CTD, and we cast that to 250 meters. It's a piece of electronic equipment that measures temperature, salinity, dissolved oxygen, pH, conductivity, fluorescence, as it's going down through the water column.
Okay, Sean, let's go down to 10 and back to the surface, please.
Roger, down to 10.
We can see that on a monitor and see how the temperature and the salinity changes with depth. In addition, we're sampling the plankton, specifically the zooplankton. So we use a very small mesh net to collect those, and we cast that down to 250 meters. When it comes up, we preserve that in a preservative so we can take it back to the lab and identify all the small critters that may be similar to what's in the salmon diet. It's kind of cool.
Yeah.